Thank you for having me here. I hope you will enjoy this little break in this workshop. Maybe you can ask, I'm going to tell about proteins, something not really related to NLP, I would say. But I hope that in the course of this presentation, you will find the link and you will see that some methods are quite similar between both worlds. So yes, I work at one of the supercomputing center in France, Idris. I'm part of the support team for both HPC and AI. And my background is in theoretical chemistry, and that's why I joined also the AI team for the support, because the chemistry is one field that's starting to use a lot AI. So that's why both expertise is quite nice. So the first thing I will tell you is I'm trying to tell you a beautiful story about proteins, and I will try to convince you that those proteins actually they are stories. If you look at this, actually in your body, proteins they have a lot of functions. They can do many things. On the left side of I can try to use that, I'm not sure. On the left side here, you have the infamous spike protein that the coronaviruses use to enter the cells, and something that it's not really pleasant, but proteins, they have other functions. For example, just on the right of this protein, you have an ion channel. And those channels, they permit ion to travel through the cells, and that's used for many things, like transmitting information in your neurons, or just equilibrate the charges in the cells. On the top, you have one of the protein that is used for moving. It's one protein that is involved in the muscles movement. Below, you have clotting factors. So if you cut yourself, you start bleeding, and those proteins will come to help the clotting of your injury. And finally, the last example is one protein that is quite fun. Actually, it's a circle that helps the DNA formation. So one thing that is really important here is that all those proteins, they have different shapes, and the shape is quite important. If you retain one thing from this talk, is that the structure of the protein defines its function in the body. And that has some implication. For example, I think that proteins, they can have mutations. Some mutations are normal, that's with the evolution, and some can cause disease. And as long you can mutate a protein without many problems, as long as you do not disrupt the structure. As long as the function is correct, you can have some mutations. So, and first thing you can see maybe that some structure they are common in those proteins. You can see maybe that we have some building blocks. For example, here we try to do that. Here we have an helix that is one of the common structure, and with a small number of those building blocks, you will have a huge variety of functions. So proteins, they are polymers of amino acids, and I will tell that the amino acid list that you have all here is kind of our token list, our word list for the proteins, for telling the stories. Usually, so an amino acid is this thing here. So we have a common building block, a chain that we call the backbone, and with the R1 here, you have a side chain that is different for each amino acid. So here you have the list, and you have several groups of amino acids. They have some different properties, and with those properties, they will be able to form the proteins. So some are charged, some are hydrophobic, some have some sulfur that will be able to bind together to form very strong bond and make some very stiff structures. So that's our list of words. I won't go into the details, but that's what we use to define our stories, our function. So one protein is, so I told you that it's a chain of amino acid. It's a sequence of amino acid. I will use this word also, sequence. And those sequences, locally, they can rearrange themselves in 3D to have those building blocks I was talking about. So we have two examples. In these examples, you see that the chain is folding on itself and to form a kind of sheet that we call a beta sheet. Another kind of structure is an alpha helix, that the one I showed you just before, that this one, the purple one here. And you have several kind of other building blocks. Not too much, 5, 6, something like that. And with all those 6, you can arrange them into 3D space to have what we call the tertiary structure for the protein. So that's the part here. And here we can start to tell about the function of the protein. But protein, they like some protein, they like to work together, and they combine into homomers or heteromers or complexes. And this is the quaternary structure, and this is also very important for the function. Okay, so that is what is a protein. If you have any question during the talk, do not hesitate to interrupt. I will answer the question. No problem. Okay. So I'm here to tell you about how we find the structure of the protein, because getting the sequence is kind of easy. I'm sure that if I tell that to some experimentalists, they will tell me no. But it's easier than finding the 3D structure. Okay, so we have experimental method to do that. Historically, we will use X-ray crystallography. For example, that's the method that was used to find the shape of DNA. So that's the picture on the bottom here. So you have those points, and with some mathematical method, you can find the position of the atoms and find the 3D structure. One thing that is very difficult with this technique is that you have to crystallize the protein. So you have to make a beautiful crystal with water or some other solvents, and that's quite difficult. To tell you, getting one structure might take 1 PhD, so 3-4 years. That's really difficult in some cases. We have other techniques to do that. We have, for example, nuclear magnetic resonance. That's the picture. Here. Here you can have either solid state or in solution. It's a bit less difficult because you do not have to crystallize, but they have this technique of also some drawbacks that you cannot use it for all the proteins. And one more recent technique is cryo-electron microscopy, and basically it's just taking picture from different angle of the protein with electrons, not photons, with electrons. In this example, you have also to freeze the protein, but not in a crystal structure, so it's a bit easier. So all these techniques are quite difficult. They require a lot of money and time to be efficient, and to be able to help. I would say the experimentalists, the theoretician, they can use some numerical methods. So you have several kind of families for the numerical methods. For example, you have all that is the sampling of the conformational space, so all the position that the atoms can take in space, and that's what is depicted here. here, you have either, so I think that this kind of picture you might be familiar because it looks like a loss landscape. I'm calling it potential energy landscape, but actually that's almost the same, and we use kind of the same methods to get to the minimum points. So to find the minimums, because that's what we want to do even for the structure, we have the sampling. So either we perform Monte Carlo simulation, so you randomly move some atoms and hope to find some minimum. Or you can use molecular dynamics, where you start from a point and you solve the Newton equation of motion with some parameterized energy functions to get to the minimums. So that's the sampling of the potential energy surface. One other thing that you can do with proteins actually is to try to find some proteins that have very similar sequence. And you suppose that if some proteins have similar sequences, the shapes should be almost the same, or at least for the parts that are very are fixed, like the helices or the beta sheets. If you have the same sequence, then you can suppose that you have the same shape. And that's called homology modeling. And basically, what you do is you take a template of a protein that you know the structure, and with the protein that you want to find the structure, you try to match the structure. And actually, AlphaFold, so what I'm going to tell about, the AI method, is kind of related to this method, to try to find some similarities in known proteins to find what we are interested in. Okay, if I want to be really complete, actually the experimental methods, they have to use some kind of these numerical methods to be able to get the structure. You need to have some refinements to get ready the structure. Okay. Any question? Okay, so the ultimate goal of AlphaFold is to start from this thing, so this list of amino acids, and to try to find the structure. And we don't know the structure at the beginning, obviously. So again, this is the spike protein, and we want to transform this what I would call story into the 3D structure. Okay, we have something that is really powerful. It's called evolution, and Darwin is helping us a lot here, because if you take several species, like I don't know, a fish or a cat, something like that, some proteins, they have the same function. So because of evolution, we have a common ancestor, and the proteins, they diverged. And it means that we have several sequences of proteins that have the same function, but they have a different sequence. You can have mutation, you can change some amino acids, you can remove some parts, you can add some other parts, and still the function is correct. And that will help us a lot, because we will be able to find kind of synonyms, if you want, in the sequences that tell almost the same story, but a bit differently. And what we do is called alignment. So we take several proteins, so sequences, and we try to find parts of the sequences that matches. And here you see that for some part of the proteins, you have a strong no mutations. You cannot have mutations, and we suppose that if we don't have mutation in this part of the protein, it means that this part is really important for the function. So if we have a mutation, it's not selected by evolution, and it's really important. But some other parts, we have more freedom. Some part, you can change some amino acid by other, it will not break the function. But if you change it by other amino acid, it will break the function. And for some other part, you will have complete freedom because it's completely flexible. Sorry. So here it's what we will use to get some kind of some sort of synonyms, if you want, for our stories. So we do it with several sequences. So we call it multiple sequence alignments, and that's one of the key ingredients of the AlphaFold model. Okay. I'm going to tell you about a competition that occurs every 2 years that is called CLSP, for Critical Assessment of Protein Structure and Prediction. And this competition, so it happens every 2 years, and what they do is to ask some experimentalists to resolve the structure, to get an experimental structure of some unknown proteins. So the experimentalists, they do not compete. The one who competes are the one with the numerical methods. And so for 2 years, they try to find the structure, they try to develop methods to get the correct structure. And at the end, you get a ranking. And here what's happened. So that's what happened since 2006. So you see, we have the GDT, so that's the score. It means that the higher it is, the closest to the real structure we are for some proteins. And you see that since 2006 until 2016, the score was around 40. To be clear, around 30, you have random structure, no match. Around 60, 70, you start to get something that is that is correct. And if you reach 90%, it's not percent, if you reach 90 of GDT, the numerical method is almost as accurate as the experimental method. So you see that we stay around the 40 here for a long time. Staying around 40 does not mean that the method do not improve. It just means that the competition is much harder every year. So the method they improve, but since the competition is harder, we stay here. And in 2018, yes, DeepMind participated in the competition with the model AlphaFold, and they gained 20 of GDT for the first time. And the next session, so 2 years after, 2020, we reach almost 90 with AlphaFold model 2. So we are getting very, very close to experimental methods with AlphaFold 2. That's a good news. Maybe you have seen that on some news saying that protein structure discovery has been solved, it's okay. In reality, it's helping a lot, but yet it's not completely solved. We have, there is a lot of work to do. Okay, so just. So I'm going to tell you a bit more about AlphaFold. I'm not really going to go too deep into the model itself because I think you have more to teach me than I can. So, but I will try to tell you what the physics here. What is the physics here? So AlphaFold was created by DeepMind. It uses TensorFlow and JAX. You can run it on CPU, TPUs, and GPUs with no problem. And it depends on several tools for getting the multiple sequence alignment, because that's one of the key information ingredient that we need. One thing that is interesting is that the data set that they used was partly self-distilled, so they used AlphaFold to get some structure that were not in the dataset, physical dataset that they were using, the PDB, Protein Data Bank. And that was one part to help improve the performance they use, so the self-distilled dataset also. So what we do, the first part of the model is to get an embedding for the sequence and the structure. So you get some information from genetic databases, and you get the MSA, so the multiple sequence alignment. You transform it into an embedding space. And you do also the same thing for templates. So we have also a database for templates. What I call templates is some known protein structure that we have in a database. And we try to find some structure that are close enough from our query to get some information about some knowledge about the structure. We have to start from somewhere, and having those information is quite important. Once you have that, you can enter so you have an embedding space for the MSA and for the pair representation. Pair representation means that we are going to, the way they represent the 3D structure is in a matrix of distances. You take all the amino acids, and all pairs of amino acids, and you compute the distance. That's not only the distance. You have other features that are implied, but basically it tells the distance between all the pairs of amino acids. And now that's the main contribution of AlphaFold, I would say, is the evoformer block. So I think that the ending with former tells you something about what is under the hood. And so the evoformer block is a sequence of action with some gated state attention for the rows and the columns in the MSR representation. And you also get some information about the 3D space with what they call triangle update. You have some self-attention also on the pairs of amino acids, but triangle because they use 3 amino acids, and they try to triangulate the amino acids. You have more information about the 3D space with that. I'm going to give you a feeling of how it works with the first part for the MSA. So what we want is to apply some attention on the columns and on the rows. On the rows, because the rows they represent the sequence. So you have each column is one of the amino acid, and at the same that you would do with a sentence, you want to have attention on the sequence, because some amino acids they will interact at a distance. It's not because some amino acids can be far away in the sequence, but very close in the 3D structure. So you need to have some attention here to get how they interact. And I told you that Darwin is helping us a lot here. We can have some synonyms for the amino acids, and that's why we also want to apply some attention on the columns, because the columns they represent some similar structures, but with different amino acids. So knowing how you can exchange some amino acid without breaking the function is very important for the model. And also you will have attention for the 3D structure. So here you will apply attention on all the pairs of all the triplets of amino acids to get information about the 3D structure. So underneath you have the model. One example of for the gated attention. I think you can recognize easily that that's a transformer model. So I'm not going to tell you more about that. If you have any question. Okay. So the next part, once we have the evoformer block, so we have one evoformer, but we are in the model. They have 48 blocks in a row with different parameters, and more than that, they use what they call recycling. It means that once you have a structure, you recycle the model at least three times to get a better and better representation, because you feed once you recycle, you feed the output here to the model, and you have some convergence at one point to a structure. Okay, now we want to build the structure, because we have nice embeddings, but we want to be able to see something. And we are going to enter the structure block. The structure block, the main contribution of AlphaFold here is what is called the invariant point attention. That's also a kind of transformer but here it will work on the geometry. And the basic idea here is to have some, they call it frames. So you have your sequence, and each amino acid is treated independently, and they can move independently in a sort of gas of amino acid, if you want. So the first part is to arrange the structure in the 3D space, but for each amino acid. And then you stick the pieces together to get the final structure. And with that, you will be able to get a structure for your protein. So it looks like this, a bit more complex, but I think that something is not very frightening for you, I think. Okay. I'm not going to tell you more about that. Oh, so yes, something important for this block. For the evoformer block, you work with the MSA, so the multiple sequence alignment. But for the structure block, you only take the representation of your input structure. But that's quite obvious, I would say. Okay. Any question? So at the end, you get some information about the structure. You get the structure. You get AlphaFold. The output of AlphaFold is a file called PDB, a format for storing proteins, and you get a structure. More of that, you will have some scoring that is called LDDT, and AlphaFold will tell you, okay, for this amino acid, I'm pretty sure that it's correct. For this amino acid, the confidence is a bit low. And you might say, okay, if the model is not very confident, it's not a good news, but we have some physics here. And you have to know that for proteins, some parts are very, very stiff. So those parts you want to have a high confidence. But other parts, they are very flexible, and it's even important for the function that those parts are flexible. And obviously, if they are flexible, you cannot ask the model to be accurate on the position, because it will vary in time. I will give you an example. When I was in master, I worked with a protein that has one of this loop here. You see the arrow written low confidence. So in the protein I was working on, you had one loop like this, and the loop has two states, open and closed. And the protein was performing a chemical reaction, and the loop was closed when you have some molecules inside that was undergoing the chemical reaction, and then the protein opens to give the product of the reaction. And so obviously the model won't be able to give you a high confidence for those structures. But that's not a problem, as long as the low confidence is for structures that are flexible, it's okay for us. Okay, so the model gives you some coordinates, but they are most of the time you will find some physical problems, some atoms that are too close, or you need some refinement. And here, what we can do, that's not mandatory, depending on what you want to have, is to perform a relaxation with a molecular dynamics code. In AlphaFold, it's OpenMM. And they will use a force field. So a force field is a way to represent the interaction between the atoms. And with this force field, you will perform an optimization of the geometry to have less clashes, to have those non-physical interaction removed. But depending on your problem, it can be enough to have the raw structure. So AlphaFold is able to do monomers. Monomers is just you have one unit. But AlphaFold is also able, since version 2.2, something like that, to perform, to study interactions between proteins. So to have those multimers complexes. And that's really powerful because most of the proteins, they will work not alone, but with some other proteins. Okay. So AlphaFold is not the only one on the market. You have Rosetta Fold. From Meta, you have ESM Fold. CollabFold, which is kind of a wrapper around several methods like AlphaFold or Rosetta, but they provide a much better way to compute the multiple sequence alignment. To tell you, they can go something like 100 faster than the method the tools that were used by AlphaFold. Okay, you go 100 times faster, but the price is that the precision is a bit lower. But it can be enough. And you have others that try to get rid of the ESM Fold, for example. One of the key differences is that they are trying to get rid of the multiple sequence alignment. Because multiple sequence alignment is something that is done on the CPU, that's just searching in database, and that's not very efficient. So getting rid of that can be very helpful. And since 2020, because AlphaFold was so powerful in the CLSP competition, many groups, they try to have their own variation of AlphaFold. For example, I worked with a team that is developing a code called Massive Fold, which is just AlphaFold, but with some more freedom in the parameters, because AlphaFold is quite. If you run the tools that are given by DeepMind, it's a bit you are a bit constrained, I would say. And if you want more freedom, because you want to have more sampling of the spaces, because the model is giving you one answer, but if you run the model with another random seed, it will give you another geometry. So that's something that might be interesting, to have this sampling. And by default, AlphaFold is not doing it very easily. So the team, they were trying to add some features to AlphaFold. And so again, we had the CLSP competition of last year. So the results were just at the beginning of the year. And still AlphaFold, or models based on AlphaFold, are still on the top of the list. Okay, so I just finished with AlphaFold, and I want to present shortly something that was done on Janzé. So Janzé is the machine at Idris. And it's a project called Ceratus. It's an open science project where the goal is to find new viruses. So what they do, it's they have some huge database with genetic information, so mainly RNA or DNA. And what they want is to screen this database to find some other some proteins that belong to viruses. And so you can see for the big numbers, 5 million samples, more than 5 million samples collected all around the world, mostly in Europe, Asia, and US. I would say North America. And that's huge, and they need to get some very powerful tools to be able to do it efficiently. So the Ceratus project was done in 2 steps. The first step is done, and what they are trying to look at is a protein called RNA-dependent RNA polymerase, and it's a protein that is very important for the viruses that are RNA viruses, because it's one of the proteins that is used to hijack the cell to produce the virus. So what they did is, the first step was to what they called high identity sequence alignment. So they tried to find some very close match for the sequences. And as I told you, if you have a lot of amino acids in common, you can think that they have the same function. And with that, with this searching, they were able to find 80,000 new viruses, and they published a Nature publication for that. That's quite impressive, but now the next step is to do the same, but with low identity. So low identity, it means that the sequence is different. And if you remember the beginning of the talk, I told you that the sequence is important, obviously, but what we want is the function. And since we can have some mutation, and viruses, they are known to have a mutation rate that is quite high, you can have the same function, but with a different sequence. And that is much more complicated to do that, because finding the same sequence is quite easy, just searching in the database. But finding the same function, so the same structure, you need to perform some analytical stuff and to find the structure. So what they did is to again screen the database, but this time they tried they had to perform the computation with AlphaFold. So they used CollabFold because it was faster. But at the end, it was AlphaFold in the back end. And with that, they found 500,000 new proteins that were close. So 500,000 doesn't mean that they found this number of new viruses, because they have to perform some more analysis to tell, okay, this is RDRP for a virus, a new virus, it's okay. And so it's an ongoing project. So that's what the first batch, and they are now doing the second batch, and they hope to find another 500,000 new RDP, so 1 million new, and try to find new viruses that will help finding cures for those viruses. So some others. It was a huge project for Janzé. They had They used 1 million GPU hours. It's quite a lot. At the time they started, 1 million was something like I don't know, something like maybe 10% of all what we have to distribute. Now we have more GPUs, so we have a bit more to give, but still it was a huge project. And they faced a problem that was not really expected. Actually, the problem they have now is the post-processing, because actually they now finding the structure is going very fast, And they have a lot of data. I think they folded, so they performed the AlphaFold folding for, I think, something like 5 million sequences. And now they have to try to analyze that. They have a lot of data. I just want to thank Johan Dufresne from Institut Pasteur. He is the one who provided me with the slides here. I think I'm done. Yes. So yes, thank you for your attention. I hope it was enjoyable.
I would like to come back to the structure that you predict. So how many? So it is an infinite dictionary of structure, if I understand correctly, and you can imagine whatever you want, or there are some constraints, and that it is a limited number of structure you want to retrieve.
I'm not sure I'm getting completely the question, but you do not. At the end, you have a few structure for them because they have different models, so the different weights for their models. And for example, with you can have at one time for 25 structures that might be close or not. You do not really have an infinite structure because you have. You do not have the constraint. You do not put constraint. You have physical constraints, but that are learned by the model. I think. I hope I answered the question, but I'm not sure.
So it's you predict some numbers, or you predict some classes, or
no, you predict the
matrix
directly the structure actually. At the end of the model, you have the structure.
Okay, so you said at the beginning, because I just don't very understand how is it represented, this structure. So you said it's a matrix of different distances between the...
Okay, sorry. So no, at the end you have just coordinates, just the atom coordinates. But for you have the embeddings of the structures, and for the MSA. And with that, at the end, you output some coordinates and this score that you have. No, you have directly the coordinates at the end.
So, in the CLSP challenge, the results are validated against non-protein structure. In the GDT is computed against that.
No. Well, yes and no. At the beginning of the competition, you have some sequences that are chosen, and some experimentalists, they will try to perform to find the structure with the experimental method, like crystallography, NMR, or something like that. And with those proteins that they found, the proteins that are not known, we do not know the structure for them. We compare it with some structure found with the numerical method. So one thing that you are sure of is that at the beginning, the proteins, they are not in the protein data bank that is used for the learning.
So yeah, the full question was, how do you go about experimentally validating the results of the protein structure prediction?
You just overlap the structure, and you compute the GDT. It's the global distance test. So you compute the distance between all the atoms. All the atoms you compare the distance of the atoms in the model and in the ground truth, the one that was resolved experimentally. And the higher the rating, the better. If you are very close, then your rating is going up.
Can I continue on that question of evaluation?
Yeah.
So I'm thinking that let's say there is some accepted structure of a protein, and then maybe you did some electron microscopy, or I don't know how you get it, but there's some way of getting ground truth, let's say.
Yeah.
And then you make a prediction. How do you account for simple things like, there might be a block move, namely you got one structure wrong, so everything after that got rotated. So in coordinate space, everything is off by a lot, but it's really one error because there was one orientation that you had wrong. So the reason I'm thinking about it is I'm thinking about what we do in speech and language. We have edit distance, but one of the things we worry about, especially in tasks like translation, is I might move an entire phrase from one part of the sentence to another, and that doesn't really mean I have 10 words wrong. It just means I have a phrase in the wrong position. And so we've struggled with metrics for evaluating this because simple edit distance, which is like, coordinate-wise differences like in that flavor, that doesn't quite explain or doesn't. In other words, two systems might be different for different reasons. One of them is just wrong and the other one is wrong by a rotation or by a mirror symmetry or something like that. So is that something that is discussed? Is that important for this task? Maybe it's not, I don't know.
Well, if you mean global rotation or translation, that is quite simple. You just align the both structure at the beginning.
Right.
You do not compare directly the output of AlphaFold and the ground truth.
Right.
You perform some rotation, translation, so that they align.
Right. But imagine there's one of these flexible structures, and then after that everything else is. In other words, you can't rotate the whole molecule, whole protein together. You'll have to rotate this part to align with the first and the other part differently to align with the second. Do you do things like that, or is that considered?
Yes, I think it's considered obvious. But at the end, if you're for parts of that are flexible, we know that they are flexible. So that's why we have the confidence, and it's taken into account. Yes, obviously. But that might be a problem because with the experimental method that are used, you have a bias because some structure you cannot resolve because they are not. For example, you cannot crystallize those. Another problem that we have, for example, I told you about the ion channel here. This ion channel actually is going through the membrane of the cell, and the environment of the protein is really important. And some of the methods cannot represent all the environment, and so you bias to something that might not be really physical. At the end, it's taken into account, but there are some problems. That's why saying that AlphaFold solved the problem completely is quite wrong, because we still have some bias. We do not have a complete view of the landscape of the protein structure. We only have, I think, we only have something like 200 million structures. That seems a lot, but actually it's not a lot. We have, with evolution, you can have different structure and something that we might not even know, actually, because we have to find the genetic information about that.
I would like to ask the question about the visualizations that you are making, because what you are getting would be just the structure, while here you are showing something very colorful. What does that mean? And does AlphaFold give you this extra information or not?
No, that's a common visualization in the field of biomolecules, because I don't have. You have just, if you want, you can just have the atoms. Maybe we have some example. I don't have to move. I think, here. Here you see another representation that what we call sticks. So the atoms, for example, here you have, I think, a nitrogen that is in blue and some carbons. And you could have the same representation for all the protein, but you do not really see well. So the depiction is, we want to see the structures. So we remove, mainly what we do is to remove the side chains and just plot the backbone in a way that represents something different. So you want to have a different representation for the helices, a different representation for the loops, a different representation for the beta sheets. So So no, that's not something that is given by AlphaFold. So that's something that is common in the field of biochemistry, because we have such huge molecules that you cannot really just depict the atoms, because you don't see anything. You just see a cloud of atoms, and it's not very useful to understand what the protein is doing.
And I wanted to ask a question regarding the project you mentioned in the end. So there is some collection that has been done mainly in the northern part of the world. Is there any initiative to fill in the gaps?
Well, I hope. I'm not really part of the project. It's just something that run on Janzai, so that's why I present it. But I'm not really part of the project, so I won't be able to really tell you what is done. But actually, the project itself is not in charge of collecting the samples. They use international databases that are fed with some samples that are collected around the world. But obviously, we have something very wrong here. The South exists also, and I think it's quite important for the viruses. We have a huge cluster in the Eastern Asia because, for example, all the coronaviruses they come from here. All the flu. So, but no, obviously there is a problem here.